All right, let's go ahead and get started because I do have a few announcements or things that I want to share. Let's see. First of all, the study guide was posted last Thursday, so it is on Moodle. I've also uploaded a couple of documents simplifying mitosis and meiosis to 1 or 2 page documents that you might want to review to help you study. Let's see. I don't know if you saw the news last week, at the end of last week, but James Watson, right, one of the scientists that made the discovery of DNA, passed away. So that was a little sad, whatever your views on James Watson are. But I thought it fit very nicely into the assignment I had to do last week and the topic that we are exploring this week. So he was a pioneer in the field, so it is sad. But he was 97, so. Let's see. If you're thinking about doing research next year, we do have a program through Gustavus geared towards first-year students called the FIRE program. There's an informative session happening on Thursday, so you can ask questions, learn more about this program. But basically, we accept a number of first-year students to do research, mostly on campus at Gustavus for the summer next year. And we prioritize first years, which is it's really hard to get research experience as a first year. So it's a good thing to go to if you're interested at all. I know not everybody in here is a first year. There is a sophomore specific program as well, the SEER program. I did the FIRE program when I was a first year. They didn't have the SEER program when I was here, but I highly recommend it. So I would say check that out. Let's see, advising day is tomorrow. If you have any questions about the biology major, there will be tabling for a couple of hours in the atrium of Nobel. Pam Kilson, who doesn't teach classes here anymore, but runs a lot of like the fellowships and students applying to grants and stuff like that. She'll be there to help answer any questions you might have about the biology major. I will note, right, organismal biology. If you are planning on taking organismal biology next semester, right, so if you're a bio major, it is needed because it's the next bio in the curriculum, right? If you are planning on taking it and you need to take it for your major, you will be able to get it. But when you look at registration, it's going to change from how it looks now. Right now, it probably says like there's 40 available seats in lecture and like maybe 16 in lab. That's going to change. So when you register, you might only see that there's 7 seats available, right? Or 10 seats available. That is on purpose, right? So we lower the caps for registration and then increase them as time goes on so that each section fills equally, right? What that means is when you go to register and you know you want to take organismal, if there's a section of lab and lecture open that you can register into, register for those sections, right? Even if it's not your first choice, register for those sections. If it's going to conflict with other classes you need to take, like absolutely need to take this semester. Chemistry, for example, if you're thinking about taking organic chemistry, right? And you can't make anything work in your schedule, there's going to be a section labeled limbo section for both lecture and lab. So if you're looking, everything's full. You can't register for anything. Register for the limbo section. What that means is after registration, I will work with you to put you in a lab and lecture section that will work, right? Now, it might not be your first choice, right? It might be inconvenient in your schedule, but we will get you into that section, right? Because we want you to take it, right? And I'm telling you this now, and I'm going to send out an email probably later today, if not tomorrow, describing all of this. But I want you not to stress out if you're looking and everything is full, right? Know that we will get you in. All right, we just have to be a little bit flexible in our schedules sometimes. All right. Any questions that I could maybe quickly answer about advising? I'd say go to advising day tomorrow. There's so many useful things and a lot of free food. So take advantage of that. Yes, there'll be a lot of places that have like free goodies. All right, so we're going to start into lecture today talking about alterations in our chromosomes. So we've talked about DNA and how it's replicated. We've talked about once it's replicated, how a cell is going to divide, whether through mitosis or meiosis. Let's talk about some abnormalities that can occur in our chromosomes, particularly that happen due to changes in how meiosis happens. All right, so if we're looking at chromosomal changes, right, there's two types of changes that we are going to look at. Changes in chromosomal structure, so how is it structured, how is it built? And changes in chromosome number, right? So how many numbers are in a pair, right? Or how many chromosomes are in each set of your DNA? Maybe there's more than there should be, maybe there's fewer than there should be. Right? It is very rare for the structure or the number of chromosomes to change within a species. Right? So say if we're just talking humans, we're just talking tulips, we're just talking rabbits, right? Within the species itself, it's very rare that these changes will occur, right? But sometimes they do, and we're going to talk about that. Now between species, right, so between humans and tulips and rabbits, differences in the chromosomal makeup and the number of them is common. In fact, that's very normal for humans to have a different number of chromosomes than a rabbit, right? Very normal. All right, we're going to talk about these variations because a lot of times they can lead to human diseases or fatalities in, like, say, children not being born because of these differences. Beyond humans, right, especially plants and organisms that have really quick lifespans, differences in chromosomal structure and sets could actually lead to the evolution of a new species. We can get a new species arising from these types of mutations, right? So it's not all bad. All right, we're going to talk about how a chromosome can change structurally first. So a chromosome, right, is normally has certain set of DNA in it, right, certain genes that are present in a certain order. We can have changes in our chromosome in that maybe a chromosome might go through a deletion event, right? Portions of our chromosome, in this case, our E gene, might just get completely deleted, right? Now we don't have that gene, right? Which is bad, especially if it's a gene that you really need. All right, so we could just delete a portion of our chromosome out. We could also duplicate a portion of our chromosome. So now instead of having one copy of a gene, we have two copies of a gene, and thus two copies of the associated allele or the variations. Right, so in this example, we have duplicated our D and our E gene, so now we have two copies of each. We could change the order of the genes, so this would be an inversion. Right, in this example, we're looking at chromosomes, or genes D and E on this chromosome. Right, it can be essentially flipped around, so now E comes before D. Right, think about crossing over. Crossing over in meiosis, we want to exchange genetic material. We want to exchange genetic material in the same genes. But if those genes are in the wrong order, we might end up exchanging different genetic material than what we are supposed to exchange. Right, so these types of changes in chromosomes we've talked about so far are just within one chromosome. Chromosomes can also interact with each other and have changes in structure. So now if we have two chromosomes, It might be that we have a translocation event, right? Where part of one chromosome is completely removed and put onto another chromosome, right? So the resulting chromosome is very different than how it should look. This can happen on one of the chromosomes, or they can be reciprocal, and that they both exchange information. So the resulting chromosomes are a little bit of each of those two chromosomes that started. Right? So here we have a little bit of each chromosome on our resulting chromosomes. All right, when I ask you, or maybe if I have a question like this on the exam, I want you to know big picture here, right? I want you to know, what does it mean for a chromosome to change its structure? What are some potential ramifications? Maybe give me an example or two, right? I would expect you to know and be able to identify what is a deletion, a duplication, or inversion. Maybe I'll use it as an example, but I'm not going to have a problem, say, where I give you a picture and say, what type of chromosomal mutation and structure has occurred here? I want us to think big picture here, right? And big picture because this is the only slide I'm going to talk about change in chromosomal structure, right? The rest of the chromosomal alterations that we are going to focus on are changes in number. All right, so know that chromosomal structure can change. And again, it is rare, but it can happen. All right, we can also get changes in chromosomal number. Like I said, between species, different species, changes in number is completely normal. All right, so humans, we have 46 chromosomes in 23 pairs. Right, dogs have 78 chromosomes. Fruit flies have 8. Potatoes have 48. Yeast have 32. Right, all very different, but very normal, right? Because we're between different species. It becomes abnormal when we're talking about changes within a singular species. Right, so when we're changing chromosome number within a singular species. All right, there's a couple of different ways we can change chromosome number. We're going to talk about aneuploidy first. So aneuploidy is variations in the number of individual chromosome types. So we're talking about within a set. Right? We're typically talking about homologous chromosomes at this point. This is where if a set should be diploid, right, and contain 2 of each chromosome, maybe one set within that genome contains 3. Right? This is how Down syndrome arises. Right? Individuals with Down syndrome have trisomy 21, which means they have an extra copy of their 21st chromosome. Right? So instead of 2 chromosomes in their 21st set, they have 3. Now, Down syndrome is called trisomy 21. Trisomy because we have one more chromosome than we should have. So we have 2n plus 1. We have 3, hence the trisomy. All right, so we can add in an extra chromosome, but it can also be the case where we are missing a chromosome. And this would be monosomy. This is where it should be diploid and have 2, but now we only have 1 chromosome in that set. Right? There's an example of this called Turner's syndrome. So in females, sex chromosomes, we should have 2 X's. Individuals with Turner syndrome only has 1 X. Right? They are missing one of their chromosomes. Now, Turner syndrome typically leads to individuals, physically it appears like they're really short in stature. Genetically, they might have problems with infertility. Right? So there are some ramifications here, but they can live a normal life. Right? Just like individuals with Down syndromes, there are some ways that it's expressed, but these individuals can live a pretty normal life. So that would be aneuploidy, changes within a set. So what happens if every single chromosome within a set were to change, right? So what if a diploid species, so we should have 2 chromosomes per set, all of a sudden turns into a triploid species, where now we have 3 in every single set, right? This would be an example of polyploidy, right? And where polyploidy becomes an issue is when it's different than what it should be, right? So if a species should be triploid, and now they're hexaploid, that's a problem. Or when a species is diploid and becomes triploid, that's a problem. Right, so even though humans, we're diploid, we have 2 chromosomes per set, typically we're showing, technically we're showing polyploidy, because we have more than 1 per set, but it's not a problem, because we're supposed to be diploid. Right, it's when diploid becomes triploid or tetraploid that polyploidy becomes a problem. All right, so we can be diploid, we can be triploid. There are species that are tetraploid. There are species that are octoploid, so have 8N. Right? We can have species like this little Botrychium fern here. I actually studied these ferns when I was at Gustavus. There's a professor that studied these. Right? They actually have 96 sets. Right? They are 96 ploid, right? Which is crazy, considering that this fern is about 2 centimeters tall, right? It's very tiny, but they have a ton of chromosomes. All right, aneuploidy in humans is usually lethal, not always. So remember, aneuploidy is when you have an additional chromosome per set or you are lacking a chromosome per set. If we look at some statistics, and I want you to remember these particular statistics. Again, think big picture here. But about 5 to 10% of all fertilized human eggs result in an embryo that have an abnormality in chromosome number. Right, so it does happen, but it says a low percentage, only like 5 to 10% of the time this happens. If we're thinking about another statistic here, about 50% of all spontaneous abortions are because of alterations in chromosome number, right? So if you lose a pregnancy, right, it could be the result of that growing fetus had an abnormality in chromosome number. But like I said, some abnormalities can survive, right? Like trisomy 21 with Down syndrome, Turner syndrome. There's also trisomy 13 and 18. Individuals with those trisomy numbers can also survive. So it's not always lethal, right? There are some individuals that can still be born. They might have some effects on their life, right? But they're still born, they are still human, they are still functioning. All right, so rare, but does happen. Let's talk about how this happens. All right, so aneuploidy in humans is the result of incorrect separation of chromosomes in meiosis, right? That we call non-disjunction. So non-disjunction happens, chromosomes are not separated properly in the anaphase stage of meiosis, either meiosis 1 or meiosis 2. So let's walk through an example here. This first example is showing non-disjunction in meiosis 1, right? And if we look, right, in meiosis 1, we should be lining up tetrads and splitting them apart. The mitotic spindle has not connected to one side of a tetrad. And because the mitotic spindle has not attached, it's not going to be able to pull apart that tetrad during anaphase. Right? So the result is that an entire tetrad moves to one side instead of being split. All right, so if we were to follow this abnormality through to meiosis 2, right, because after meiosis 1 we get 2 daughter cells, each of those go through meiosis 2. One of those cells is going to have an extra chromosome, right? And that chromosome, if it gets split normally again during anaphase, will result in cells that have an extra chromosome, right? They have trisomy. The other cell is lacking a chromosome because it wasn't split evenly, right? When that cell goes through meiosis 2, the resulting gamete is going to be missing a chromosome, right? So we're changing the number of chromosomes within a set, right? So this typically happens within like 1, maybe 2 sets during non-disjunction here. All right, so this can happen in meiosis 1, but maybe meiosis 1 happens normally, right? Non-disjunction does not occur, a splitting of tetrads happen, everything is going fine until meiosis 2. So maybe we have non-disjunction in meiosis 2. So after meiosis 1, we have 2 normal cells, right? One of these cells is going to split normally, right? Non-disjunction is not going to occur. It's going to split normally and result in gametes that have the proper number of chromosomes. But one of these cells has non-disjunction. Again, the mitotic spindle did not properly connect to one of our chromosomes, and thus the sister chromatids did not split in meiosis 2, resulting in one of these gametes having an extra chromosome and one of them lacking in a chromosome. All right, so we can have it can mess up how our chromosomes are split, and that's how we get these gametes that are either lacking in a chromosome or adding in a chromosome that can go through the process of fertilization. All right, so now let's switch our mind to polyploidy. This is where the entire set has been duplicated. So instead of a diploid organism, you're now a triploid organism. In humans, polyploidy is lethal, right? It's 100% lethal, right? Polyploidy of the fetus is a really common cause of losing a pregnancy during your first trimester. Again, it's rare. It's about 2 to 3% of pregnancies have this occur. Right, causes about 15% of miscarriages. Sometimes, if we're talking about triploids in humans, right, triploidy individuals can be live born, but they will die shortly after. It is 100% lethal. And then tetraploids can also form, right? But they will die really early in pregnancy. Right? So 100% lethal. Not good for humans. Now there's a lot of text on this slide. Yeah?
Can't you live if you have mosaicism, triploidy?
Mosaicism? I have not heard of that.
That's where only some of this also. There's a 69 and some of them is the regular.
Oh. I think I have heard of those cases. I haven't put a name to it. Yeah, so if maybe some of the cells are triploid, there is a case where you can live. It's when all of the cells have triploidy that it's lethal. And I know that's the case, and I think it happens in like a specific organ. Yeah. All right, polyploidy in other species, though, common, very common. Right, so fish, for example, are often tetraploid. Yeast are tetraploid. Strawberries are octoploid. Let's see, frogs can be tetraploid. Plants can be hexaploid, triploid, tetraploid. right? It's actually really common in plants to have polyploidy, right? In fact, it's very common in plants to have tetraploidy, or excuse me, polyploidy. Right? Not tetraploidy, part of polyploidy. Right? And in fact, polyploidy in plants has been linked back to how different species of plants have arisen over time. Right? So they've led to speciation events, right? So the developing of angiosperms, which are flowering plants, the development of ferns. If we look at all the vascular plants on Earth, right? So that's pretty much all plants except for mosses, right? About 70% of them are believed to be polyploidy, and to have developed polyploidy fairly recently in their evolutionary history. Right? Polyploidy in plants can actually be a beneficial thing. Right? They can lead to plants that can tolerate harsher environments, and plants that maybe even have the ability to self-pollinate, so they don't have to rely on pollinators. Right? There's a lot of advantages for plants. So I threw up a map here by biome and by ecoregion, but basically if you see a lot of red, the redder the area, the more common it is for plants to be polyploid, right? It is super common. It is actually pretty beneficial. There's actually a field of biology, I worked in a lab once where somebody was doing this, that you can extract out the DNA from plants and then separate out all of the individual chromosomes and count them up to see what ploidy level the plant is you're studying. It's very tedious work. All right, I want you to put this into practice here. I want you to assume that all of these individuals should be diploid to start with. I want you to determine, is this an example of aneuploidy or polyploidy? I'll give you a couple of minutes here. I don't have the timer on the screen.
So the first one is aneuploidy, and it's monosomy. This is the non-monosomy, monosomy, astenosome, and
How many stickers or how many did you put up?
we covered a lot of the library. So every Twilight book in the library has one. There's also one beneath every Twilight, so if you pull it out, you'll see it, and if you read it, you'll find one. We put it where Jasper first shows it. Just kind of stuck in places. I'm covering this building right up in my head.
Good thing we're prioritizing.
I don't have lab today.
I know, isn't it great?
Yeah, which means I don't have any other classes.
Okay. Oh, not interesting.
What was the most interesting thing you had to do for bio? We took the DNA that has jellyfish blood and we placed it into a cells.
I think it's just like this.
Yes. We also had to dissect it.
Yeah.
And mine, for sure, but it didn't stick, so it was already like half a year. It was the worst experience of my life.
Between the X and the Y, I did two total college tours. One of them, they were dissecting a cell rupture, and the other one, they pulled up.
This is not like a set.
Yes! When I was in anatomy, we had
This one? Yeah, so it should be a diploid
when I
individual.
was in anatomy, I dissected a cat.
So in this case, there are
So
multiple instances of aneuploidy.
my cat had a heart condition that caused their heart to
I mean, there's so
go
many that
always
you
different.
could
But
argue
you can
that
only figure out if you have polyglobin.
Yeah.
So
Kind
they
of like
killed
in
them.
between.
Yeah.
They were greatly preserved, though. It was amazing.
Well, what do they all
Yeah.
have to be like? your argument is they all have to see this, they all have four, like there's extra sentence.
I really hope they do, because I want to just see this.
Yeah, isn't that four for all of them? Those ones apply to it? When it
No,
has one
one's
extra
in my
or one
UT
last.
class, they brought in sheet ones and a sheet.
Yeah.
It's not like a full set.
So it's like
Not exactly simulated diversity, but whatever.
Okay, so what's the one more time? Gross
That I would think.
Then you have
Fewer than there should be or more than there should be within a singular set. Versus polyploidy is where there's more or fewer every single set. Or at least most of it.
So like the argument we're making with this one is she's saying that some children, when they like a triploid where they're all with a couple less than, or when it
I've also eaten.
Yeah.
That makes sense. I just had, I had camel cheese. That was pretty good.
All right, let's go over these karyotypes that I have on the screen here. So like I said, we're going to assume that these individuals should all be diploid. Let's start with this one on the bottom here. Is this an example of aneuploidy or polyploidy? Aneuploidy, because some chromosomes have an extra one, right? So if we were to look at, there's my timer. This one actually has several sets that have 3 instead of 2, right? But it's not every single set that has 3. Because it's just some of the sets, it is still aneuploidy, right? Now, likely, well, only one of these is human, right, if we're counting out the number of chromosomes. I was going to say if this was a human, likely this individual probably would not survive, but this is not a human because we only have 19 sets of chromosomes. So this is a different organism. All right, what about the one on the top? Aneuploidy. Why do we say aneuploidy?
It's missing
It's missing one. Which one is it missing?
the Y.
An X or a Y. All right, so it's missing one. Is it trisomy or monosomy? Monosomy, because it only has one where it should have 2. Right, and this one is in the sex chromosomes. Right, there either should be another X or there should be a Y. Right, but there should be 2 total, and there's only 1. Right, so this is an example of aneuploidy. What about the one in the corner down here? Yeah, this would be an example of polyploidy. If this individual or if this organism was supposed to be diploid, almost every single one of the sets is now a tetraploid. Now there's a few in here that only have 3 instead of 4, but the majority all have 4 chromosomes instead of 2. Right? That is an example of polyploidy. It's affecting every single chromosome set. All right. Any questions before we move on? I'm going to take that as a no. All right, so we're going to move on into our last topic that we have before the exam next week. Right, so all the slides that I have posted today, those are all the slides that there are going to be before the exam. Now, we're not going to get through all of them, so whatever we don't get through, we'll get through on Thursday, and there'll be some time for exam prep on Thursday. Okay? But this is our last topic. So we talked about DNA replication. We talked about cells that divide after DNA replication. But what we haven't talked about yet is how do we use this DNA to make a functional product. And that is what we're going to start talking about here with gene expression, with transcription and translation. All right, so first of all, we're going to define the word we are going to define the word gene expression, but I forgot I put in this review here. All right, we're going to review some terminology because this terminology is important and it's going to also be important in the next unit as well, so we need to know it. All right, so if we're talking about chromosomes, right, this is a singular chromosome, this blue one. That black one is a singular chromosome as well. Maybe the blue one is the chromosome from your dad, and the black one is the chromosome from your mom. right? Because they contain the same type of genetic information, they're a homologous pair. All right, if our chromosomes look like this, right, we still have a singular chromosome, right? Singular chromosome representing the information inherited from dad, and then a singular chromosome representing information inherited from mom. The difference is that these chromosomes have gone through DNA replication, right? So there's two copies. Those two copies are still joined together, right? So we still have a homologous pair. Right? The difference is our homologous pair now contains sister chromatids. All right, so our DNA would look like this after the S phase of the cell cycle, when a cell is duplicating its DNA and getting ready to go through mitosis or meiosis. All right, let's go back to the singular chromosome without the sister chromatids. On our chromosomes, on both of them, remember they're a homologous pair, they have the gene for eye color. All right, so the gene is that particular genetic sequence, right? It is the stretch of nucleotide base pairs in that region of our chromosome that encodes for eye color. Right? Once this has been transcribed and translated, which we'll talk about in a bit here, the result is a functional product. Now, because one chromosome came from mom and one chromosome came from dad, you could have two variations on this allele, or on this gene, and we would call that an allele. Right? One of two versions of DNA sequence at a given loci, given location. Right? And I know we've already introduced some of these terms, but review is always good. All right, so if we look at the variation here, we're looking at the gene for eye color. We might say on the chromosome from dad, we have the B allele, the capital B allele. Now, we haven't talked about recessive or dominant genes yet. That'll be next unit, right? But this one is a dominant gene, meaning if it exists, it's the one that's going to show up. And it encodes for brown eyes. Whereas mom has a different allele that encodes for blue eyes and is a recessive allele. Right? So if you have these two chromosomes in your DNA, right? You have two different alleles. It's actually going to be the one, the brown eyes that you will actually inherit, because, like I said, that's the dominant trait. If it's present, it's going to be expressed. Right, a little bit preview into next unit here, but I think it made sense to talk about it in terms of the context here. All right. Let's put these terms into practice before we move on again, and I'll give you a couple minutes.
Those are lovely.
I like it. We have this down. We used to have, in the old set of curriculums in the labs, we used to have a lab dedicated to learning these terms and going through mitosis and meiosis. But we don't have that anymore. What was that? I mean, it was helpful, but in my opinion, it's a boring lab. Because you're working with beads, you're not doing like an experiment. A lot of them has, but some of them, it's been really on your high school.
I took a JP class.
Yeah. So for you, a refresher is probably not as needed as for some other people.
Some people are less dedicated to the ground.
Maybe less dedicated, maybe have less access to the cost.
That's the idea.
Okay.
Yeah. Yeah, we had biology.
On the phone, this one they said.
Well, I had to.
Oh, see, we had 5 different biology guesses, and I was like, I'm gonna take it all.
See, I did too.
Yeah, there was no option.
It's called, the school started hiring different teachers. They hired, and then completely. You guys started. Yes? Yes and no. Me and my siblings went to this one. That's $30 for each of us. And then we also together.
After a while, it's just
All right, let's fill in these blanks. So let's start with the blank on top. Blank on top is pointing to this chromosome within the pair, as well as this within the pair. What term would I put in that blank up top? Chromosome. I actually said it because I'm so used to talking about them as chromosomes. Right? They would be a chromosome. They're both called a chromosome, even though they look slightly different. Right? I don't think that's my next one that pops up. But let's talk about why they're different. All right? The one that looks like an X has 2 sides that are identical. And what would I name this? Sister chromatids, yes. And then each set, right, so either this set or this set would be labeled as a homologous pair. Yes. All right, so within each homologous pair, right, we have a region that is the same. What I call this region? It's a gene, right? It's a particular set of DNA encoding for a gene. All right, so if I have 2 different versions of that gene, what would I say it is? An allele. Nice. I know this might seem tedious, but this terminology is going to come up so much. All right. So now let's talk about gene expression and what gene expression means. All right, so if we think about our cells, every cell in our body has the full complement of DNA. Right? Every cell in our body has 46 chromosomes in 23 pairs. But if you think about all the cells in our body, right, they don't all look the same, even though they have the same DNA. They don't look the same because portions of that DNA are being expressed and portions are not being expressed. Right? So if we say, look at these columnar epithelial cells, typically they're found around like your intestinal lining, right? They look very different than a nerve cell. They also perform different functions than a nerve cell, right? Intestinal cells have to do with like digestion and absorption of nutrients. Nerve cells is like processing information, right? They have different functions. Now both of these have the same DNA in their nucleus, right? These have 46 chromosomes, but they're only going to turn on the genes that are actually needed. So think of it as like a light switch. If you are in a particular room, you'll turn the light switch on because you need light there. If you're not in a room, right, you might keep the light switch off to conserve energy. Right? So a nerve cell is going to express genes that a columnar epithelial cell might not express. Even though they have the DNA for all the genes, they're only using a portion of the genes encoded within the DNA. All right, so we're only using a portion of the genetic sequence. A portion is being expressed for each individual cell. Right, so hence the gene expression. What genes are actually being expressed, what genes are being turned on. Right, we can look at gene expression at a couple of different levels, right? And these levels are intertwined with each other. They relate to each other. We can look at gene function on the molecular level, like the molecular function of a protein product, right? So a gene is going to encode for a particular protein that will be made that relates to that gene. So we could say, what is the function of that protein? Or we could look at the gene and say, what is the trait that is conferred by the gene? And maybe that trait is the function, right? But maybe it's different. So let's look through an example. Here we have a chromosome, and on that chromosome is the gene for flower color, right? So we have this DNA sequence that relates to flower color. This DNA sequence, once it is expressed, will translate into a protein. That protein has a molecular function, and the function of that protein is to make pigment, right? So this protein makes pigments, right? That gives the organism its trait of having purple flowers, right? So the trait is having purple flowers. The function is making pigments that will be purple, right? So they're related. And if we think about proteins have a ton of functions, right? They can be enzymes. They can be molecules that help transport, think like hemoglobin helps transport oxygen in our blood. They can be hormones like insulin. They can help our muscles contract. In other organisms, maybe it's a protein that helps produce silk, like if you're a spider, right? Proteins have so many functions, right? And the function that it performs translates into that organism's trait. All right, well, how do we go from DNA sequence to protein? Well, that is the center of our conversation here. It is called the central dogma of biology, right? You'll hear this throughout probably every single one of your classes that you will take in biology, right? It's a huge deal. It's how we go from our genetic sequence, right? The A's, the G's, the C's, the T's, and make a product that actually functions in our body. Make protein. All right, so when we're talking about the central dogma of biology, we're talking about transcription, which is the process of taking DNA and making an RNA copy, and then translating that RNA copy, or translation is our next step, taking that RNA copy, translating it into a protein that has a function. Right? So the process of transcription and translation, that's how genes are expressed. That's how genes are turned on or off. That's like the light switch that you are turning on and off when you enter a room. Right? If you turn that light switch on, you're going to take your DNA sequence, turn it into RNA, make a protein. All right, so the ultimate goal is to make a protein. We're going to look at the first step. Right? The first step is simply to make an RNA molecule from the DNA. Any idea why we need to make RNA? Why can't we just use the DNA directly to make a protein? Exactly. It has to be able to travel outside of the nucleus. So DNA stays in the nucleus. RNA has the ability to travel outside of the nucleus. And outside of the nucleus is where the proteins are going to be made. Right? So we have to have this intermediate molecule that can travel from the nucleus into the cytosol and carry the information needed in order to make a protein. All right, so if we're talking about a gene and we're talking about transcription, there's a couple of regions that we need to identify within a gene, right? So if we're looking at a particular sequence of DNA, there's all sorts of regions, right? We have what's called the regulatory region, right? And that's a region that provides a site for proteins to bind, right? So these proteins that bind to the regulatory region help regulate transcription. So it might tell it when to start, how much to make, how fast should it be made, right? A bunch of regulatory proteins. We're looking at that image, we see it's right at the beginning of this section of DNA that we're looking at. Right, now within this regulatory region, we also have a region called the promoter. And this is simply a sequence of DNA that indicates that this is where we need to start transcription. This is where we need to start making our RNA molecule. It's kind of similar to when we talked about DNA replication, how primase made the primer, and the primer was the spot that indicated to the DNA polymerase where to start. Same concept here, right? Except this is a sequence within the DNA itself that says start here. We look at the other end of this sequence, we have this termination region. Right? So termination region is where we are going to terminate this process happening. So this is where DNA transcription ends. Right? It's a sequence of the DNA that says this is where I stop. All right, so we have these sequences that help regulate transcription, right? The promoter region, where we're binding regulatory proteins, the promoter region, which says start here, and the terminator region that says end here. All right, now between the promoter and the terminator region, right, this is our coding region. Right? This is the region that we're actually going to use to make an RNA molecule. This is the gene. Right? This is the gene that gives eye color or hair color, right? This is where that trait is going to be located. Right? So even though we have this regulatory region, we have the terminator region, it's still our genetic code, but it doesn't really code for a gene. It just helps transcription start and end. All right, but you might notice within this region, we also have some more terms. All right, within the coding region, we have exons and we have introns. Exons are regions of the DNA that are going to be kept in the final RNA product. Right? These are the regions we want. These are the regions we are interested in. These are the regions that are expressed, right? So they are expressed, they are the exons. We also have regions that are going to be transcribed, right? So they're going to be in the RNA, but they're going to be taken out of the RNA before the RNA is used to make a protein. We call these introns, right? Intervening regions. And we see in our image here, we see introns kind of intervening in between our exons. Right, so we're eventually going to remove these introns. Now, introns themselves can have functions. It's an area of ongoing research. If you're interested in it, I would say do some research on your own to see what the current research says, right? There are still scientists trying to figure out why do we have these if we're not using these. Right? What is their function? All right, let's do a review. I'll give you a couple of minutes.
So gene expression is okay. Several regions the regular score, the promoter, and then I love that picture.
Like never seen it.
I haven't either. I don't know why. I'll give you the right.
The robot, like the guns and that wheelbase.
I've also never seen it.
I like that starts to talk. So you started. Do you know what you're getting your father?
binds proteins that help
Do
determine.
you know the game Wingspan?
So there's sort of actions.
We're getting every one of the packs of cards.
Okay.
Oh,
We have all the X-Men, right?
It only makes sense. How are you coming on your holiday?
It's like a
that's
cell.
what gene expression is. It's like choosing which genes are being expressed.
You buy for.
I have to figure out.
We're gonna have.
I only buy presents for one set of my parents.
That's the one that we don't have.
I feel bad there is one grandma keeps getting me fun things. She's trying, but I'm not gonna get her husband.
That's where it begins, and then
My parents don't think it's
Well, you just found out one of my cousins is
oh yeah, so now she's like, that's the kind of excitement.
All right, if I were to ask you, what is gene expression? What would you tell me? How would you describe gene expression to say your parents who are not biologists? Yeah, what gene is expressed? What do I mean by expressed? Yeah. So it determines what a cell is going to do, right? So it's basically like whether a gene is used or not, whether a gene is turned on or off, right? So if it's turned on, it has a function, it is working, it is performing. All right, so all genes are all genes in DNA expressed in every cell? No. We have all the DNA, but they're not all expressed, only the ones that are needed. All right, and if we're thinking about this genetic code, right, there's several regions that aid in transcription. Okay? And I have this nice little diagram up here. So if I were to ask you what is the region at the beginning of the sequence, what would you tell me? Promoter region. You could also say it's a regulatory region. And the promoter is actually within the regulatory region. All right, what does the regulatory region do? What is its function? Yeah, it allows the site for proteins to bind, and that those proteins help regulate transcription. How fast, how much, when should this gene be expressed? And the promoter region, what does the promoter region do? Yeah, this is the cue to say this is where we start transcription. All right, on the opposite end, at the end of this segment of DNA, what region would we find? The terminator region, yes. And what does the terminator do? It terminates, yes. So I'm not talking Arnold Schwarzenegger here, but it ends when transcription is going to happen. And the region in between, what could we call that? What was that? Yeah, we could call it the coding region. We could call it the gene. All right, so that's the region that's actually going to be turned into RNA. All right, any questions before we move on? All right, so we've kind of learned a little bit about the gene, but we haven't yet learned how that DNA sequence turns into RNA. And that is where we are going next. All right, so we're going to look at all the steps, or I should say a simplified version of the steps of transcription. How do we go from a sequence of DNA nucleotides to an RNA strand, right? That is the process of transcription. Right, we are making an RNA copy of the DNA that stays in the nucleus. And it's that RNA copy that will leave the nucleus and be used to make a protein. All right, so by the time we're done talking about transcription, you can go back and look at this image and know exactly what we are talking about. All right, so if we're talking transcription, we're going to talk about 3 stages, right? Transcription has 3 stages: the initiation, the elongation, and the termination stage. All right, now we're going to start with the initiation stage because that is the first stage. All right, as I talk about these stages, we're mostly going to focus on how transcription is done in bacteria, but I'm also going to compare it to how this is done to eukaryotes, like our cells. Now, the reason we're focusing on bacteria is because it's simpler, right, and gets you started thinking about how transcription happens. Eukaryotes is a lot more complex and has a lot going on, and is beyond the scope of what I would want you to really understand for Bio 110. Now, as you move up in biology, you'll start to look at this in more depth. All right, so let's start with bacteria. Right? Bacteria in the initiation phase. The first thing that happens is that this protein called the sigma factor is going to find the promoter region. Right? So in this image, the sigma factor here is this smaller protein. It finds this promoter region that is in blue here. All right, once the sigma factor finds that promoter region, then our RNA polymerase, right, so another protein, is going to come in and bind to the sigma factor. Right, so we have our RNA polymerase. It's binding to the sigma factor that has already found that promoter site. Now, the job of the RNA polymerase is to open up the DNA sequence and make the new RNA strand. Right? It has a lot of jobs. It's going to unwind the DNA, right, just in the section that it's bound to. And that's what happens in the initiation section. As we move on to elongation, we'll see how this same protein then makes the RNA strand. Right? So in eukaryotes and bacteria, so prokaryotes, this initiation stage is completed when the strands separate near the promoter region, and that area that is separated we call an open complex. So it's similar to a replication bubble, where it's a region where the DNA is unwound. Right, the replication bubble in DNA replication, right? DNA replication happens radiating out from both sides of this replication bubble, and that bubble stays open. Right, this open complex is only going to move in one direction. Right, so we're only going to have replication moving in one direction. Or I should say transcription happening in one direction. And once the RNA molecule moves on, right, the DNA that was open is going to rewind again. Right, so we're just opening it up. Now, when we open up this section of DNA, right, we have 2 strands that we've split those hydrogen bonds apart. One strand is called the coding strand. It can also be called the non-template strand. There's two different names you could put to that. And another strand is called the template strand. So the coding strand is actually not going to be used, right? It's the template strand that this RNA polymerase is going to read and build an RNA molecule off of. Now, it may sound confusing. Why is this called the coding strand if that's not the strand that I'm coding, right? Well, it's called the coding strand because essentially it's going to be the same as the RNA strand that will be produced, except the RNA strand has uracil instead of thymine and a different sugar in the nucleotides. Right, so we're only reading off of the template strand. Now, in eukaryotes, like I said, it's more complicated. I really don't expect you to know this process in eukaryotes. It's very similar, except there's way more proteins involved. Right, so the same basic steps happen, except there are way more transcription factors than just the sigma factor. And there's way more RNA polymerase types than just the one found in bacteria. Right? But again, big picture, all of this leads to us finding the promoter region and starting to replicate our DNA into RNA. All right. So we have this open complex. We need to make the RNA. Right? And this is where RNA polymerase kind of does all of the heavy lifting, right? It's going to synthesize an RNA molecule. The direction in which it synthesizes is important, right? So again, we're talking directionality of our strands. RNA polymerase synthesizes or builds the RNA strand in the 5-3 direction. It's using our complementary base pair rule, except RNA does not contain thymine. It uses uracil instead. All right, so it's going along. It's adding in the complementary nucleotides. And I do have a visual after the content, or not the content check, but after the question slide that I'll present. Also happening here is we are releasing any proteins that we no longer need. Right, that sigma factor that found the promoter region is released. Right, we don't need it anymore. Same thing in eukaryotes. All those proteins that helped start our transcription is released because we simply don't need it anymore. Right, and once those are released, this RNA polymerase just kind of slides on down the line. Right, so it's going to read. It's going to read. It's going to build. It's going to move on. It's going to read. It's going to build. It's going to move on. And notice again, it's only reading one of those DNA strands. It's only reading the template strand. The other strand, it's unwound, and it's just kind of hanging out. We're not doing anything with it. All right, so we've initiated transcription. We are elongating our RNA strand that we are making. And eventually, right, we're going to get a strand of RNA made during this elongation phase. So again, directionality is important. It's only made in that 5-3 direction. As the RNA polymerase moves, it's going to rewind once it's read that part of the DNA. All right, so this visual on the side here, we have our RNA polymerase. It's kind of like this triangle molecule over the DNA. We see we have a green region up front, right? That would be the promoter region. We have this red region towards the right, right? That is our terminator region. And we have this region in between that we are going to read. The RNA polymerase forms this open complex and reads the template strand and keeps moving, right, in the correct direction. And as it moves, the DNA rewinds behind it, and this RNA strand grows. Right? right? So as we're reading it, we're adding to this DNA or this RNA strand, and this RNA strand is growing. All right, so this happens until the RNA polymerase hits that terminator region. Right, once it hits that termination region, it's going to fall away from the DNA. It's no longer going to be bound to the DNA. And the RNA transcript is also going to be freed from the RNA polymerase and from the original DNA itself. So you can see in the image here we have 3 distinct features. We have the DNA that it just read. We have the RNA polymerase no longer attached to the DNA. And we have this RNA strand, again, no longer attached to the DNA, no longer attached to the RNA polymerase. All right, so at this point, this is the end of reading the DNA. It is not necessarily the end of transcription yet, especially for eukaryotes. All right, I'm going to allude to that, but we're going to do a check-in here, and then we're going to watch that visual to help you kind of visualize what's happening.
Strand, I guess, yeah.
And then elongation keeps on. It's like sigma's gone. The alpha of the sigma. It's just manipulation. It's like round. The protein is
Understanding it simpler than some of the other processes.
Yeah, definitely simpler, but definitely complex, you think?
Yeah, yeah.
Oh, I love it.
Yes. Are we ready for advising day?
Yes, yeah.
Yeah.
We, I had, so, like, I think what we did, I don't know if it's for everybody, but our peer model, we have one for our peer model on top of, so we have to meet with our peer model before we meet with our advisors, so like, and he's a biology major, so I was like, oh,
Very
it makes it easier for me.
Yes.
So I was like, he was just like, yeah, you look at something at ours, and it's like, that's just gonna be like my advisor, or like my, what's it called?
Yes.
There it
Yes.
is. My teacher for that is a math teacher, so it's not gonna really know. But yeah, you have to get that done.
Yeah, but yeah.
So it's like, you know, just talking about it, like, all right, looks good.
Okay.
Yeah.
And then once you like say you declare a biology major, then you'll get a biology advisor.
Right.
Yeah.
Would you recommend taking?
I wouldn't have to. I read that options. I would.
The elective course.
You get to choose either that one or the chem one.
Is it evolution, ecology, or behavior? Or
Yeah.
Okay. Choose that or the chem one.
My aunt just got out. Which is crazy.
Ah.
That's like
For biochem.
14 years younger than any of my other cousins.
It depends on what side of biochemistry you're interested in. If you're more interested in biology side,
I was like,
how
dead
things
set
work and
in
translate and how organisms work.
and then she just had to go.
That'd be nice. But if you're interested more like the chemistry side or like the more details of things, then maybe take the chemistry course. Yeah.
So it's like a preference, isn't it?
Yeah.
She's the cutest
Yeah.
baby.
They're both like the same difficulty you would say.
Like the cutest newborn. Even like ugly. Like regular.
Yes. I would say you don't have to make that decision until next year.
Okay.
Because I know both courses have prerequisites that you'll complete this year. And at that point, you'll have a biochem
The
advisor.
baby looks like a daredevil.
And they'll get to know you and be able to
Like
advise
set
you
right on.
which one to take. All right, so we're looking at transcription. The first stage is initiation. What happens here? What binds to the promoter region? The sigma factor, yes. And then what binds to the sigma factor? Yes, RNA polymerase. And what does the RNA polymerase do? It's going to pull the strand apart in this step and form an open complex. Yes. All right. So we're starting. Our next step is elongation. What does RNA polymerase do in this step? Yeah, it reads off the template strand and makes that RNA. Yeah, we're building our RNA strand. Right? Any proteins that initially bound in the initiation step to start this process will be released, including the sigma factor. And the DNA, once it's read, once that template strand is read, is going to coil back up into a helix, right, as the RNA polymerase moves along. All right, so we come to the end, our termination step. What is happening here? It's terminating, right? It's ending. So once we reach that sequence, the mRNA, and the RNA polymerase, they dissociate. Right? They are no longer attached to the DNA. All right, so what is the name of the protein used in transcription for the bulk of transcription for bacteria? RNA polymerase, yes. And then in eukaryotes, there's multiple of them. All right, any questions? All right, like I said, this is not the end of transcription, particularly for eukaryotes. All right, let's watch a visual of what has happened so far. Now, I couldn't directly embed this visual in the PowerPoint slide because of this video's copyright protection, but let me move to the correct area here. I did link it. If you downloaded the PowerPoint early, you might have to go back into the folder, and it'll be on a slide, a link to this video. Let's watch a visual. We'll get to that. And I'm going to pause it there because that's all the further we've talked about. All right, like I said, I linked that in the slides if you want to go back and watch the whole thing. But I alluded to transcription is not done yet, right? So for bacteria, it actually is done. But eukaryotes, because we have to make everything more complicated, there's more that needs to happen to that RNA strand that we just made, right? So eukaryotic mRNAs have to be further processed. There's 3 different ways it's processed, right? One way is that we have to get rid of those introns. Remember that we have exons and introns. Exons are what we are concerned about. That's where it carries the genetic code for the trait that we're conferring. Introns are those intervening regions. They've been transcribed, but we don't want to keep them in our mRNA. We're actually going to splice them out. We're going to remove them. All right, so it's going to go through a process where we are splicing out these introns, so that all that is left are the exons, right? The exons that are in RNA format. All right, so we're removing those introns. There's a couple of other things that need to happen for this RNA to be mature, to be full and ready to move on to the next step. We're going to add a cap, and we're going to add a tail. All right, so we've spliced out these introns. All that's left is the exons. We're now going to make a couple more modifications, like adding in a cap. Right, so at the 5' end of our RNA strand that was just made, we're going to add a cap of a modified guanine. All right, so it's this guanine that has an extra methyl group. It's really similar to the nucleotide that has the nitrogenous base guanine, right? But it's slightly modified. We need to add in this cap to allow the RNA to exit the nucleus, right? In order to exit the nucleus and bind to the ribosome, that's going to be the area of our next stage translation. Right, so we're adding in this cap to the 5' end. Right, we're making our mRNA. So mRNA stands for messenger RNA. And we call it the mRNA, the messenger RNA, because it's the messenger between the DNA sequence and the ribosome that's actually going to make the protein. But we're also adding on this tail, and we call it a poly-A tail, right? Because essentially it's a sequence of a couple of hundred adenines. Right? These adenine nucleotides are added to the 3' end, and the reason is that we want to increase the stability of the molecule. We don't want it to break down on us, right? And we want to increase its lifespan. Right? So RNA molecules, we know they are not as stable as DNA. Right? They don't have as long of a lifespan, but they need to exist long enough for it to get to a ribosome and be translated into a protein. Right, now, even though we're adding in a bunch of adenines, right, the code for adenines was not in that template strand of the DNA that we read. Right, so it's not encoded in the gene sequence, but rather it's added afterwards. Right, so if I was to look at the gene and the DNA, and I were to look at all the base pairs, I would not have a section that tells me to add on 200 adenines. Right? That's not in the DNA. It's added after the fact. All right, let's do a review.
So we're going to get rid of the they don't need them. And then they're going to put a little hat on it with the tail.
I haven't been until today.
If you were
So I haven't.
do some mRNA, what would be the hat that you wear?
Oh, I think it's a traditional top hat. I think that's
That's what I was gonna say.
Too bad you don't get like a cane or a monocle to go with it.
I have a cane.
Is it a fun cane or a bad cane?
It's made out of wood.
That's a good way to visualize it.
It's like freeform, it's not like
I bet they need sisters do that if they're big.
Oh, I'm sure they do. Okay, does RNA processing occur in prokaryotes?
oh, no.
Actually, we should.
Yeah.
I just didn't see it.
Don't need it.
The remove
Don't need that.
concepts that.
They don't know how to do things.
So
Why would they
Splicing.
care?
Yep.
Exactly.
Just destroying everything. Drag around with big lump.
What molecule is mRNA? Messenger RNA, and then there's like what? They lost rRNA.
What are office hours?
Normally they're on Wednesday, but
MTR
like it's
sounds
by
like
today, so everything's
So NPR, but with a different.
Transit. What's NPR stand for? National Public Radio?
Yeah.
those office hours. I can pencil in for time, we just gotta course.
Yeah, I don't know when to get my mom.
Last year I got a funny
I
message.
anticipate my Thursday office hours.
That's going to go.
I hope well attended.
My mom,
Yes, because it's the last office hours before the
we
exam.
need to get rid of them.
What time exactly?
I dog on her like, I'm like, Mom, you have like,
Noon to 2:30, so I would like to get my office.
I'm like, Mother, you have like $100. There's no goddamn way you need all of this. She's like, I know, but I like them. And then she goes and buys 5 more.
What?
This was
All right, I have given you a picture of a gene, and I have asked you how is this gene, once it's turned into RNA, going to be modified in a eukaryote? What is one way? Get rid of the introns. We have to go through splicing. What else? We have to add in a cap, that modified guanine cap, so it can leave the nucleus. And last thing, adding in a poly A tail to increase its longevity once it enters into the cytosol of the cell. All right, so essentially we're taking our DNA sequence, we're making RNA, we're splicing out the introns, we're adding a cap, we're adding a tail. All right, does RNA processing occur in prokaryotes? No, only in eukaryotes. And what molecule is leaving the transcription stage and moving to the translation stage? mRNA, yeah. Maybe the answer was more obvious than you thought it was going to be. All right, so mRNA now moves into our next stage, right? And our next stage is going to be translation. It's where we take this strand and translate it into a sequence of amino acids and build our protein, right? I'm actually going to end here because this is where my last class ended. I will stick around though, so if you have any questions or want me to draw anything out on the board, I'm happy to answer them.